To this webinar. I am Marilla Bowen, Product Development and TCS Engineer for Alstom Mougeot. Today, I'm pleased to do this presentation with Morty Wasavi, VP of Strategy and Business Development at SISCAPA. So today we will talk about the advantage of our fiber-based solution for the driver spot testing, and Morty will present you how SISCAPA assay allow identification and protein analysis quantitation in human blood specimens. To make sure this webinar runs smoothly, I will kindly ask you to put your microphone on mute, and if you have questions during this presentation, please use the chat box in the GoToMeeting menu, and we will answer to your question at the end of this presentation. So first, let's start with a little background about Alstom Mougeot. So we are a global leader in manufacturing and supplying sustainable and innovative fiber solutions to customers all over the world. We are more than 8,000 employees brought to work for more than 7,000 customers in over 100 countries. And we have 45 plants and converting sites in 14 countries with a global presence of sales office. And our annual net sales is about 3 billion. So what we do in Alstom Mougeot, we offer custom-made fiber-based materials. Fibers are the core of what we do. They are the core denominator of our products and solutions for everyday life applications. With the pictures of these slides, you can see that we serve various markets with a wide range of products. We have compostable food packaging or beverage. We have fire filter media in transportations, materials for home buildings and furniture. And our medical business unit provides solutions for protective fabrics with face masks, surgical drapes, and gowns. And we also have solutions for diagnostics with two key products, the component of rapid diagnostics kits and the card for the biosamples collection, which is the purpose of this webinar. So what are our solutions to support the diagnostics markets? First, let's start with the components of the IVD device. Please, can you put your microphone on mute? Thank you. So with the component of IVD device, Alstom Mougeot has an experience in this market since the 80s, and we continue to lead this industry by providing high-quality fiber-based material for rapid kit tests. We have a wide range of conjugate and absorbent pads, media for blood separation, for lateral fluorescein, or for dipstick devices like the pregnancy test. And the second category are the specimen collection cards. We offer simple and cost-effective solutions for everything related to ambient preservation of the biosamples. Alstom Mougeot is a key leader in dry blood spots. We manufacture 2 of the 3 paper grades used for the newborn screening. As the DBS gains increasing importance thanks to its strong advantage compared to conventional blood collections, we propose various solutions with a portfolio inspired by the customer experience and their feedback. So the dry blood spots technology has been for more than 50 years and is used in different fields of applications. And in the recent years, DBS gained increasing interest, extensively used in healthcare, especially in challenging environments with the area with limited resources. This DBS technology leads to simplification of blood collection process and the reduction of the cost involved. First, with an easy collection, you don't need particular skills to make the sampling. You can do home sampling. And as it's non-invasive, it's a less painful method. DBS is stable at ambient temperature, so you simplify the transport. You don't need cold chain boxes. You have no biohazardous risk, so you can use regular mail and you have no breakable specimen. Thanks to the ambient preservation, you have a cost-effective storage without need of freezers or particular equipment, so space and cost saving with low energy consumption. And with the DBS, you have a fast recovery, a short time processing with simple extraction, or you can use it with a direct punch in direct elutions. And with the DBS, with dry blood spots, you get reliable analysis with an automated workflow to get quantitative and qualitative analysis. So what kind of solution we can propose for infectious diagnosis? During this particular period with the rapid spread of the COVID-19 across the world, this led to an increase in the demand of healthcare goods in general, and in particular, protective medical products, medical devices, diagnostics, and screening tools. As we saw, the DBS is a convenient tool to enable diagnostic testing for viral disease due to transport, handling, and logistic advantage over the conventional venous blood sampling. The benefits of this method has led the World Health Organization to recommend the dry blood spot for HIV and hepatitis B and C diagnosis. So let's have a look to those data for the suitability of our fiber-based solution for the diagnostics of HIV and hepatitis infections. And let's start with the HIV. So DBS is suitable for testing antibodies, antigen, or nucleic acid, and this technology allows the population studies or is program monitoring. Ashram Mungjo is a leader in this market segment by selling millions of biosamples cards and 5-cycle cards, especially in Africa. Here on the table, you have different methods listed of the test from the DBS for HIV infections. First, you have the viral load measurements to detect virological failure during antiretroviral therapy and modify it in a timely manner. You have the testing for HIV antibodies to help the early detection of HIV infection, for example, during pregnancy. This allows the early initiation of ART and reduce the mother-to-child transmission of HIV during pregnancy. You have the HIV detection. The DBS facilitated the detection of HIV by DNA PCR in resource-limited countries. With our cards are suitable for manual and automated HIV detection with the good stability of nucleic acid during the storage under different conditions. And the last method is the HIV drug resistance. You have a monitoring survey of HIV patients under ART, and the DBS collected on our card show really good performance for HIV-DR detections. By speaking of performance, let's have a look to the data from the publication listed on the left bottom of this slide. So this publication mentioned our card for their study. And on this graph here, you have data for the HIV RNA testing on a DBS in a routine clinical setting. So on the first graph, this represents the correlation between plasma and DBS viral load measurements. And on this graph, the dashed line is the virological failure, and the solid line is the slope 1, displaying perfect concordance and correlation of 0.95. You can see some aligned dots at the bottom of this graph. Those dots are due to undetectable viral load measurements. On the second graph, this represents the analysis to evaluate the concordance between plasma and dry blood spot viral load measurements. The solid line on this graph is the mean differences between plasma and dry blood spot viral load. And you can see that you get very strong correlation and good concordance between plasma and dry blood spot viral load measurements. Now let's have a look to HCV infection. The diagnostics of HCV infection is made by detection of antibodies, antigen, and genome of the virus in the serum samples you can obtain with blood collections. So for that, DBS is an excellent alternative for HCV diagnostics since it is less invasive and it can be stored without refrigeration, and trained personnel is not required for the prelevement. So regarding the viral load measurements, a study shows that HCV viral load measurement in plasma in venous DBS and in capillary DBS gave similar results with correlation always exceeding 0.99. And a good concordance between plasma and venous DBS quantification, as well as between plasma and capillary DBS viral quantifications. You also have enzyme immunoassay designed to detect HCV core antigen and antibodies to improve the early detection of HCV infections when you have molecular diagnostic method not available. And for this type of method, a study shows good stability among various storage conditions. So on the table of this slide, there are results for sensitivity and specificity of HCV antibodies and antigen on the DBS by storage conditions. So you have different storage conditions. You have minus 80 degrees for the gold standard, 4 degrees for the refrigeration rather than freezing, 21 degrees for the storage at room temperature, 37 degrees for the storage in high temperature environment, and 37.4 degrees you have 3 days of alternating 12 hours at 37 degrees and 12 hours at 4 degrees to simulate variable temperature during the transport. So on the result, you can see for HCV antibodies were detected from the DBS in all HCV infected patients in all conditions, except for one patient in whom the antibody were not detected from the DBS for the one store at 37 and 37 degrees, 4 degrees. On the second part of this table, you have the results about temperature affected the core antigen detection. And you can see that the storage at minus 80 and 4 degrees, you had the highest sensitivity at 94.1%. And you can see that the differences in sensibility between conditions were in a minimal range from 91 to 94%. When the analysis is performed using the quantification limit of 10 femtomole per liter, the detection range was from 80 to 85%. So you can see that the storage at different temperature do not affect the sensitivity of your methods. Now let's have a look to HPV. Persistent infection with human papillomavirus causes cervical, anal, and genital cancer. Consequently, you get HPV DNA testing to develop, which has been developed as one of the primary cervical cancer screening methods in several countries. And most current HPV testing methods are based on a liquid sample carrier, which is limited by the large volume samples, the risk of leakage, and challenges for the storage and transportation, especially in remote rural areas. With our generalist cards, we provide an alternative sampling medium for the sample collections. For the moment, we don't have published data which make a direct comparison of generalist cards and liquid sampling medium using the HPV testing. But the Department of Immunology, Genetics and Pathology of Uppsala, the Uppsala University in Sweden, tested our cards for HPV testing by comparing performance of these cards with the competitor cards using the real-time PCR. For that, they spot different concentration of HPV plasmid and human DNA. And they made the HPV recovery by real-time PCR. So this assay detects and quantifies the human gene and the HPV16. And here on this slide, you have the result of the PCR showing the total of measured copy of the DNA from the paper punched from the center of 1 microliters of HPV plasmid and 1 microliters of the human DNA. You can see on this graph that you get higher recovery performances from generalist cards than our competitor for the HPV plasmid and for the human DNA control. This shows the effectiveness in the direct use of our cards with self-collected samples in PCR for the molecular detections. For HPV detection, you can also use the method of HPV genotyping, which is based on amplification technique and which allows the detection of the viral DNA of HPV genotype. And this could help to monitor the change in the viral load over time. So we saw the usefulness of the DBS in diagnostics, but we can also propose the long-term preservation of your biospecimens at ambient temperature without treated card. Here the example is for the GenSaver cards. So to check the quality of the ambient preservation of DNA, we made an accelerating aging study on dry blood on a human blood spot spotted on our GenSaver card, and we put it in an accelerating aging by using the standard ASTM 9080, which provides a guideline to establish a protocol of accelerating aging. So we put the dry blood spot at this accelerating aging, and at different time we made the DNA extraction, and we quantify the DNA by Tachman with the Tachman by qPCR with Tachman chemistry. So on this graph, you can see that we have high reproducible extraction yield for until 22 years. And you can see that between 15 and 20 years, you have a drop of only 30%, which show good preservation of your DNA over time. You can use this GenSaver cards also for the STR profile. And to check the quality of the STR profile, we made a graph by representing the number of validated loci and the average PHR, which shows the good quality of the STR profile. As you can see, you get sufficient and good quality of DNA to provide high quality STR even after 20 years of storage at ambient temperature. After 20 years of storage, you get complete profiles and an average PHR up to 85%, which show a really good quality. And if you put your punch and paper directly in the mix to make a direct amplification, you will get 100% concordance with a profile made based with the extracted DNA. So now I'm at the end of this presentation, and now I will hand over to Morty.
Hello, everybody. Thanks, Mary-Laure, and thank you for the invitation. Allow me to share my screen. Okay, so I hope everybody sees my screen now. Please let me know if there are any technical difficulties. I would like to start by thanking Ahlstrom for putting this webinar together. It's obviously interesting times for the minds of the field to get together and brainstorm about how we might be able to use a DBS-based technology to tackle one of the most severe health issues that we've seen in our lifetimes. So on that note, I'm going to be talking about the SISCAPA DBS platform. SISCAPA is a patented technology worldwide, and it has been in the business of clinical diagnostics by mass spectrometry for more than 10 years. The work on dry blood spots started for us about 6 years ago, and the work that I'll be describing today is a work that has particularly focused on different inflammatory diseases and their impact on the proteins that allow you to measure inflammation in human body. So just to give you an overview of the field of proteomics in diagnostics in general, there are 2,000 to 5,000 proteins by the latest counts that have been detectable by different proteomics measures. Interestingly, only a fraction of these have been in clinical use. So if you do a survey of the proteins that have been approved by the FDA or have gone under the regulatory aspects to be used clinically, there is a list of about 230 of them. About half of those, 117 of them, have been approved for IVD in vitro diagnostic tests by the FDA, and about half or 110 of them are tests that are laboratory developed tests. So basically you need to send a sample to some sort of reference lab to be able to measure these proteins. Out of the list of about 230 or so, 50 of these proteins are regularly used by almost every hospital in the world. And a great deal of these proteins relate to the issue of inflammation in the body. So we in SISCAPA have focused on replicating assays for these proteins so that the SISCAPA equivalent for these assays exists. So as for how a clinical diagnostic test is run on proteins right now, the 51 tests that I mentioned, you can see the list on the right-hand side panel here. The proteins against which there's a green dot are proteins for which we have an equivalent SISCAPA assay. But the vast majority of protein biomarkers are measured with immunoassays. In fact, more than 80% of clinical proteomics uses immunoassay as a platform for measuring proteins. And this is an interesting market because with immunoassays and the platforms that exist, you don't have the capability for multiplexing. So if you wanted to measure all of these proteins at once in a given sample, the incremental cost is simply prohibitive. When you look at protein quantitation in clinical settings, the issue that you quickly run into is that the most interesting biomarkers are usually low abundance, and therefore you need a sensitive method to be able to measure them. But more importantly, the matrices that are used in clinical settings are often complex matrices. So you have human plasma, whole blood, CSF, and so on and so forth. So there are three issues that comes into play when you're implementing a protein biomarker in clinical settings. The first one, sensitivity for obvious reasons. The second issue is the issue of throughput, because for a lot of these proteins, you need to be able to go from the acquiring of the specimen to reporting the results in less than a day. And the third issue is multiplexability, and this is a cost factor. As I mentioned, with immunoassays, multiplexing is either absent or increasingly limited. And as a result of that, the cost to run these platforms is simply too high. So there are two approaches to clinical proteomics. Immunoassays I have mentioned. A small fraction of proteins are measured using LC-MS platforms. And these are straight liquid chromatography that are usually more than 30 minutes per sample, very low throughput, but highly specific tests. Both of these approaches are fraught with problems, and that's where SISCAPA come into play. So when you think of SISCAPA, by way of background, you basically think of a hybrid approach. In an immunoassay, you have two antibodies, one for capturing the antigen you're interested in, and the second one for detecting. So there are two antibodies that bind to a given antigen, and that gives you an indirect measurement of the analyte that you're interested in. With mass spectrometry, straight LC-MS, you don't use any antibodies. The advantage of mass spectrometry is that you get unequivocal direct identification of your analyte using the mass spectrometer. The problem is that in a mass spectrometer, you do not have the needed sensitivity to look at the low abundance biomarkers. And secondly, the throughput is extremely low, and therefore you cannot implement this on a regular basis in clinical settings. SISCAPA is a hybrid approach in that it uses a single antibody which is specific to a proteotypic peptide from the protein biomarker that you're interested in. And that antibody is enriching the target that you're interested in. The second antibody of an immunoassay is replaced by a mass spectrometer in the case of SISCAPA. So one antibody to enrich the target, the secondary antibody is replaced by the mass spectrometer. In doing so, we basically take the advantages of a mass spec assay and an immunoassay and leave their disadvantages behind. To give you a little bit of a deeper understanding of the SISCAPA workflow, there are three main steps that are involved in the workflow itself. The first step is the digestion of the sample, and the sample is typically a blood sample. And the enzyme that we normally use is trypsin. The second step is the enrichment of the target peptide as well as an internal standard. I should mention about this internal standard. For every SISCAPA assay, there is a stable isotope standard that is produced, which is identical in sequence to the target you're measuring, but obviously is differentially labeled. And because of that, it can be differentiated by a mass spectrometer. This internal standard, aside from quality control, is added for quantitation purposes. So the second step is the enrichment of the target as well as the internal standard using the SISCAPA antibodies. Followed by a proprietary wash and elution step, which effectively eliminates completely the non-specific background and only enriches the targets you're interested in. And the final step is the measurement of the target peptide as well as the internal standard using a mass spectrometer. That ratio, the peak area ratio, then gives you a measure of the amount of the analyte that you had in the particular sample that you were interested in. Because of the peptide enrichment using the antibody, the sensitivity compared to traditional mass spec methods is increased by 10 to the power of 3 on most cases. So to give you a ballpark, in a typical LC-MS workflow, you're measuring proteins in the microgram per mil range in the plasma. With SISCAPA, we typically measure biomarkers in the upper picogram per mil and easily in the nanogram per mil range. The second issue, which is when you enrich this target, you're depleting the non-specific background, directly allows you to increase the throughput of the assay, because now you don't need a long liquid chromatography to separate these peptides, because essentially you're getting a pure sample. So we have demonstrated in the past that in some cases you can eliminate liquid chromatography altogether and simply inject your eluate directly onto the mass spectrometer. And so the assays that we measure, they typically extend a dynamic range of more than 10 to the power of 6. This workflow, I should mention, is fully automated on various platforms. These are simple liquid handling platforms that are commercially available. And secondly, I should mention that the workflow is designed to be scalable with the idea of being able to generate big data when it comes to proteomics applications. There are only two components in a SISCAPA assay. There is the anti-peptide antibody components, and these are antibodies that go through a proprietary manufacturing process to meet the SISCAPA standards, and the second component is the corresponding internal standard peptide. Now, this is important because we talked about the issue of multiplexing these assays. With the SISCAPA platform, you can simply add and remove components for a given protein without jeopardizing anything in the performance of the assay. So because of the use of mass spectrometer in this technology, multiplexing is something that's easily achieved. We have demonstrated that you can go as high as a 50-plex or higher without jeopardizing the performance of the assays. Having said that, in clinical settings, it is rare that people actually run a 50-plex assay. We typically see that multiplex panels are run in the range of 2 to 5-plex. The test is already deployed as LDT in various reference labs in the United States and Canada. The first SISCAPA test to be used clinically is a test for thyroglobulin, which is a biomarker, a protein biomarker for measuring recurrence of thyroid cancer. This is a test that's typically available as an immunoassay, but the problem with immunoassays is because you have an indirect measurement, you don't really see what you measure, and there is a possibility of false positives and false negatives. In the case of thyroid cancer patients, 30% of these patients develop autoantibodies against thyroglobulin, and therefore there is a false negative rate of 25 to 30% in people who have cancer. In the case of SISCAPA, you don't have this problem because you start the process by digesting the entire sample, and that destroys all the protein-protein interactions. And therefore, this is a highly specific test that reference labs like Mayo Clinic use to report the recurrence of thyroid cancer. Now, the SISCAPA application first started by measuring these proteins in liquid samples, and that's what the thyroglobulin example was. About 5 or 6 years ago, we started applying this technology to dry blood spots. And obviously, the potential here is huge. You have the obvious advantage of remote sampling and reducing the cost of sample transfer, which is important from a clinical perspective. But importantly, with reducing the costs, what this allows you to do is to increase the frequency of sampling. And this was something that nobody had looked at in the past, which is if you have these biomarkers and their reference range for a population is known, what happens if you start tracking these biomarkers on a personal level? So instead of referring to a population reference range, what happens if you establish a personal reference range for each of these protein biomarkers that are of interest? And so we started exploring that question and its relationship to gathering mobile health contextual information, for example, dietary input or exercise or any kind of medication, so on and so forth, and try to demonstrate the link between your everyday context of life versus the changes it would cause in your biomarkers. So before I delve into that problem, I just wanted to make a point about collection devices in general, as far as dry blood spots is concerned. There are, to our account, there are more than 20 different companies that are making some sort of a collection device which allow you to do remote sampling. These companies are primarily focused on the user friendliness of these devices, so the ease of use. And they are, for obvious reasons, focused on volumetric control, because in a dried sample, if you don't know the volume of blood and the composition of the blood in terms of hematocrit, there is no way you can accurately measure protein biomarkers. You can still measure small molecules or DNA and RNA with some success, but when it comes to proteomics, you absolutely need to know the volume of sample and the hematocrit before you can compare individuals and report protein concentrations. What these vendors have unfortunately missed, and I say this after trying the devices from these vendors, is the need for automating the sample processing when it actually gets in the lab. Everybody is familiar with the traditional way of collecting dried blood spots, for example, in neonatal screening, and you have a tech sitting in the lab punching these samples as they come in. It's a process that have been implemented for decades, but given that there's low frequency of DBS use in clinical settings, nobody has really thought about automating the process of processing DBS samples when it gets in the lab. So in our case, the story is different because what we are interested in is increasing the frequency of sampling as much as possible. And so we want an automated way to be able to handle the samples when they get into the lab. So with that in mind, we started working with Ahlstrom to design a new SISCAPA-specific collection device. And what this is a collection device that is designed in a diary format. So essentially, every day you can collect a sample. There are currently in this format, there are 4 pages of this. On each page, there are 8 fingers. These 8 fingers correspond to a column of a 96-well microtiter plate. So essentially, when this device gets in the lab, the papers get separated, and they fit right into the 96-well microtiter plate without the need for punching the sample. So a fully automated process, amenable to further automation as we continue to improve the design of this device. So this is how the device looks. This is a prototype of it. It's the dimensions of it is the size of a credit card. The cards are separated by plastic, and when you actually apply the sample, you simply lay the card flat, which comes in a box with a device to measure humidity and basically components to reduce humidity in the sample as much as possible. You do a simple finger prick and you collect the sample. Now, another thing I should mention, because of the added sensitivity that we get using the SISCAPA technology, we are interested in reducing the amount of blood that's required to do these analyses, which is sort of contrary to where everybody else is going in the field of microsampling. Everybody's interested in collecting a couple of hundred microliters of samples. In our case, we can do our process in the range of 1 to 10 microliters of samples. So the lancet that we use to collect blood is identical lancet to what diabetics use to measure their glucose on a daily basis. And therefore, it's extremely user-friendly. And so far, the feedback that we've had from the field is extremely positive. So on that note, the first question that comes into mind when you're measuring proteins in a dried sample is whether or not your results are parallel with what you would otherwise be measuring in a venipuncture liquid sample. What we call this is a parallelism question. And so here, what we have demonstrated for a list of 27 different proteins is that when you measure these proteins in whole blood versus plasma, and ultimately in dried versus liquid, the results for vast majority of the proteins is absolutely parallel. So there is no difference between measuring these proteins in capillary blood versus venous blood, whole blood versus plasma, or dried versus liquid. So those are the three components of variability. The outliers that you see on this graph are there for a good reason. So hemoglobin, obviously the protein in red blood cells, you don't expect to see much of it in plasma. L-plastin is a protein in white blood cells, and myeloperoxidase is a protein in neutrophils. So these cell-based proteins obviously cannot be on the parallelism line because you don't expect to see them in plasma, but you do see them in whole blood. Haptoglobin is an interesting protein because it is a protein that tightly binds to hemoglobin when the red blood cells leak and burst while you're prepping your plasma sample, for example. And that connection between haptoglobin and hemoglobin, the affinity between the two of them is highest affinity known in terms of protein-protein interaction. So haptoglobin is off the chart because in this prep, when you're prepping this, you're lysing red blood cells, haptoglobin binds to it, and therefore the amount you're measuring in plasma is going to be off the parallelism line a little bit. So the gist of this graph is that you can measure these proteins in capillary dried blood, and the results would be equivalent to venipuncture liquid blood. We divide our assays into different panels when it comes to dried blood spots particularly. There is the inflammation panel that I'll be talking about today. This is the list of proteins, most of which already in clinical use to measure inflammation for various disease conditions. For example, C-reactive protein is one of the most used protein biomarkers in clinical diagnostics. But we do have other panels that we use in dried format. There is a new panel that we've been working on. This is the iron metabolism panel. So it turns out that, for example, if you're iron deficient, the reason for iron deficiency cannot be deduced simply by measuring ferritin. So by using a panel, multiplex panel of proteins, not only you can determine if a person is iron deficient, but you can also determine what is the likely source of that iron deficiency. We do have a cardiovascular risk panel. an oncology panel, and what we call the LDX wellness panel. Because when it comes to collecting regular DBS samples, it is not only the question of differentiating between health versus disease. The question really changes from whether or not you're normal to whether or not you're optimized. So you may not have any clinical symptoms, but are you living to the best potential afforded to you by your genomics? That's the question we're trying to answer with this wellness panel. So the question of volumetric control and normalization came up. In our case, we don't need to control the volume of sample and the hematocrit at the time of application of the sample, which is what a lot of device vendors are focused on. In our case, we use a panel of proteins, and namely these are albumin, IgM, and hemopexin. And these proteins by mass, they constitute more than 80% of your plasma proteome. And by measuring these proteins, what we realize is that these proteins are incredibly constant in a given individual over time. And secondly, by measuring these proteins, you're effectively by mass measuring how much plasma there is in the particular amount of DBS sample that you had in your system. So using this normalization strategy, the volumetric variation in a punch of DBS or in a card of DBS is about 15%. Using this strategy, you can reduce that volumetric variation to 3%, and that allows for high precision tracking of these proteins in a given individual. So the two graphs here, they demonstrate how by normalizing you can reduce the noise that is introduced in a DBS system because of the volume. So you can simply look at albumin, I think, is the best example. There's a lot of noise if you don't correct for volume, but that noise goes away when you do correct for it. The bumps that you see on the lower parts of these panels, those are actual inflammatory responses. And that's something that I'm going to discuss in just a second. When we started doing this work about 6 years ago, the very first experiment that we did, we observed something that was really, really interesting, and that is the idea of having a protein fingerprint. So although we are measuring the exact same peptide in a list of 8 different individuals in this case, for 2 proteins, immunoglobulin M and hemopexin, these are the same peptides coming from the same gene, but the absolute levels of these proteins is very different in the individuals. This is something we refer to as a protein fingerprint. So your protein fingerprint is going to be different than everybody else's. We did a study, which I don't have a slide for right now, but we did a study in identical twins, where the genomic part of it is theoretically identical between the 2 individuals, but the proteins show an absolutely different protein fingerprint between these individuals. So you can see that in one example that is highlighted here, there are 411 samples from an individual collected over 9 years in dried blood spots. You can see that these proteins are very stable, and you can see that that person differs from everybody else. And so this idea of a protein fingerprint is what we're pursuing in this type of dense sampling. I'm going to be talking about our inflammation panel. Inflammation, can be caused by a variety of different disease conditions. And the rule of thumb is that increased inflammation is bad, and you should be moving towards reducing inflammation as much as you can. This is obviously very non-specific. If you're measuring CRP as an indication of inflammation, you see that CRP is usually elevated in all of these inflammatory responses. So the question becomes, what is the point in measuring CRP at all? And the point is, if you longitudinally measure inflammatory proteins and then you connect it to the context of an individual's life, then you have some sort of an actionable intelligence based upon which you can improve the health of that individual. So the panel that we use for tracking inflammation in dry blood spots is listed here. The panel is a panel of acute phase reactants. These are proteins that, by and large, they are increased in their concentrations when you have an inflammation, with the exception of proteins like albumin, which is a negative acute phase reactant. It has a decreased level when you're severely infected with something, for example. The proteins that we measure are for innate immunity. Part of the innate immunity is what we measure. We measure adaptive immunity by measuring immunoglobulin M, and we measure cellular response by measuring myeloperoxidase, which is the protein in neutrophils, the first line of defense. When you look at the individual baselines for these proteins, you find something interesting. So in blue is cases where CRP and serum amyloid A and LPS binding protein, these inflammatory proteins are increased as a result of a disease. So for example, a cold, a viral infection, or pneumonia, a bacterial infection. In orange, you have those proteins when the person is not reporting a health status. So they're going about their day as if nothing is happening. But interestingly, there are some fluctuations in this baseline. And when we first saw that data, we thought that, okay, that must be the noise from the assay. But then we started looking at the correlation between CRP and serum amyloid A, for example, and we saw that they correlate very well. So even in the baseline, when CRP is increased, you see that serum amyloid A is increased by the same amount, or the same pattern, I should say. So by multiplexing these assays and measuring multiple proteins, we realized that there is at all times a baseline inflammatory response as you go about your everyday life. Now, to the human body at macro level, you don't notice these inflammatory responses, but they are there at molecular level, ready to trigger a massive response if you contract something. So looking at this multiplex, multi-level tracking of inflammation, here's a case of an individual with food poisoning. The very first response that you get in an individual is a cellular response. So the very first protein that increases on day 1 of food poisoning, this is, before the person actually has any symptoms, is myeloperoxidase. There's a spike in it, and it goes back to the baseline levels. On day 2 to 7, when the person is actually feeling ill, that's when your innate immunity kicks in. These are proteins like C-reactive protein and serum amyloid A. And you see that there's a massive response, 200-fold in the case of SAA, higher than the normal baseline for that individual as a result of food poisoning. Interestingly, from day 6 to 20, that's when acquired immunity kicks in. At this point, the person doesn't necessarily feel ill. They have recovered from the food poisoning, but that's when your immunoglobulin M is starting to produce. And you're developing immunity against a particular infectious agent that caused that episode of food poisoning. We have done this for a variety of different inflammatory responses. So you can see that this is normalized according to that person's baseline. When you have something as simple as a flu vaccine, you do get a tiny little response, and that's very fortunate. But you compare that to something that, like, for example, when you have pneumonia versus an upper respiratory infection, so this would be a simple nose cold, you can see the difference is 10 orders, a simple order of magnitude, so 10-fold increase in the response of these inflammatory responses. If you have a simple cold, these proteins increase about 100-fold from your baseline. But if you have a bacterial infection, like pneumonia or a kidney infection, the response can increase more than 1,000-fold from your personal baseline. Here's a case of tracking these inflammatory proteins after a hip replacement surgery. So you can see that the person was collecting DBS samples before surgery, and that's their baseline. They're experiencing pain, and that's why they're getting a hip replacement. As soon as the surgery is done, there's a massive increase of about 100-fold in inflammatory proteins, and gradually the response decreases. The points on the graph that are highlighted with an asterisk, there's 3 of them. Those points are points where the patient was reporting some sort of pain. And that reporting of pain correlates perfectly with a spike in their inflammatory response. At the end of the process, by about day 60 or so after surgery, they now have a new baseline. So you can see that the new baseline for inflammation is twofold less than the baseline before they had the hip replacement surgery. So this is a case of a successful hip replacement surgery, and as a result of it, inflammation in the body goes down. Again, on this case, we are correlating the little spikes that you see with the pain medication that the individual was taking. So this would be an example of linking your protein biomarker data with your contextual information, which in this case is the amount of medicine, pain medicine that you take. And you see that there's a spike in pain medication that you take every time there's a spike in your inflammatory protein response. So in this individual, this is a very dedicated individual who's been collecting samples for us for a very long time. You can see that you can model the inflammatory response as a result of surgery. This is what we call a personal loop model. So you start basically on the axis, you have your SAA response versus haptoglobin, which is another inflammatory protein. And the numbers that you see on the chart, they're days after surgery. So you have day 0, that's when the person started. And you can see the loop is forming. So by about day 3 or 4, you can see that the person is on their path to recovery. They're on their path to go back to square one. In the case of an inflammatory response that's going wrong, you don't complete this loop. So basically, inflammation keeps going, increasing and increasing in a linear form until a catastrophic clinical event takes place. In this case, you can see that the loop completes and the person goes back to normal. What is immensely interesting is that for this person, we also had samples for a cold. So you can see that in a nose cold, again, you have a loop. You start from day 0 and then you feel ill. By about day 4, we can predict that this person is on their path to recovery. And in this case, it takes about 2 weeks before they're fully recovered. But interestingly, using the same exact proteins, because we know the context and because we know the baseline for that person, you can differentiate between a simple nose cold versus surgery and other inflammatory events. So in this case, the person had a significant upper respiratory infection as well, much more severe than the cold that they experienced. So you can see that they have a much broader loop, much more severe loop. But again, fortunately, by about day 9, you can see that the person is starting to recover. So this is what we call actionable intelligence and personalizing your protein biomarkers. Is when you have your baseline, you can have these kinds of personal health models when it comes to these protein biomarkers. This is an example of a person with Crohn's disease, and I like this slide because it shows you the importance of increased frequency of sampling. So this person was the first individual that started collecting dried blood spot samples for us on a daily basis for about 6 months or so. So I'm demonstrating the difference between if we were looking at the protein biomarkers on a weekly basis, which is a top panel, versus the daily basis. So in this case, you can see that if you measure these proteins on a daily basis, you can track changes that are otherwise missed if you were sampling on a weekly basis. So there are 3 events, Crohn's attack events that we detected in this individual, and we do have the context in terms of what the person is eating, how much water they're drinking, how much alcohol consumption there are, and it turns out that the 3 cases of Crohn's attack, they correlate perfectly to 3 weekends when the person was out and about, drinking a lot of alcohol and being severely dehydrated. And you can compare the Crohn's attack response to the food poisoning, and this person had a food poisoning case, and the difference in response. So you have a personal model for this person that allows them to see how the context in their life correlates with their biomarkers. So this person is in remission. Haptoglobin, which is a measure of gastrointestinal inflammation, has been on the decline over the 2 years or so that we've been tracking this person. So he's been trying all sorts of things from increased vitamin D to increased probiotics, and clearly they're working, as you can see in the longitudinal tracking of one inflammatory protein. Cystatin C is a protein for kidney function. You can see that the kidney function is in the case of an individual who is aging over time, from over the course of 8 years, this person is aging. Presumably the kidney function is deteriorating, and you can track that using a protein called cystatin C. So what we have observed is that these proteins, they change on a daily basis. In some cases, for example, for research or clinical trials, it makes sense to do daily sampling and attach that to the contextual information from that person. In other cases, you can track these on a longitudinal basis over a much longer time period of weeks to years. So bringing all of this into the current perspective, the inflammation panel I just described is being used in a major hospital in the United States right now to track COVID-19 in frontline nurses in that hospital who are dealing with COVID patients. The idea there is you want to know if the person is obviously infected, and there are qPCR type tests that allow you to test whether or not the person has the SARS-CoV-2 virus. But you want to know the severity of infection in these nurses, and importantly, whether or not they're recovered before you allow them back into circulation. And the way you can do this is by establishing the baseline for these nurses before they're exposed to these COVID patients, track them as they're treating the COVID patients, and ultimately, if they get sick with the virus, you can track their recovery. There is another way by which we are applying the SISCAPA technology to the pandemic. We are developing SISCAPA assays against particular antigens on the virus. So I'm sure you've heard on the news about the various antibody tests that are being developed and how they're not the perfect test. We are going after developing a SISCAPA antigen test that directly measures viral proteins, and we're hoping to have that test ready within the next 3 months or so. So at the moment, globally, we're looking for partners who either have access to retrospective samples collected from people who have contracted this virus, or partners, particularly in pharma, who are looking to run clinical trials, perhaps in terms of developing a vaccine or looking at the effects of a particular treatment on this virus. So on that note, I'm going to acknowledge our partners, both in pharma and in the clinics, and as well as our academic collaborators. And this is the core of the SISCAPA team. Our business end is located in the United States, led by our CEO, the inventor of the technology, Dr. Lee Anderson. And our R&D facilities is located on Vancouver Island on the west coast of Canada, and that's where I'm located. And if there are any questions, I'd be more than glad to answer them.
Thank you, Morty, for this really interesting presentation.
My pleasure.
So we have time to answer to a few questions, so please feel free to ask, or you can add your question in the chat box. So if nobody have questions, we will stop this this session. I would like to thank you, Morty. Yes.
It was my pleasure. And if any questions come along, please do not hesitate to contact me. you can visit our website at www.siscapa.com, and there's a way to contact us through that website. Thank you.
Thank you, Marty. Thank you, everybody, to attend to this presentation.
Thank you.
Thank you. Bye.